Alright, so this video is going to be about osmosis, and if you haven't already, stop this video, go back and watch the diffusion video so that you'd watch that first before you watch this one, so this one will make more sense. In this video, I'm going to talk first a little bit about why osmosis and osmolarity is important, and then I'm going to talk about how osmosis works, and at the very end, I'm going to say a few words about our experiment for this week for osmosis. So why is osmosis so important? Well, it's the movement of water, and all living things require water because life itself evolved in salty oceans. And so all the chemical reactions that life depends on to occur best in a salty environment. And not just any salty environment, but a specific concentration of salts. So as life moved from those oceans where it was fairly easy to maintain those conditions onto land, they had to carry a little bit of ocean with them. And so every cell in your body and every cell of every organism on land carries a little bit of ocean with them inside their cells in order for the reactions of life to occur properly. So understanding what affects osmosis and how water moves across membranes is important for understanding how life works. So let's get to what osmosis is. Osmosis is the diffusion of water down its concentration gradient. And having watched the diffusion video, that's pretty straightforward, right? However, this is a special case of diffusion. And it's special because it's a situation where we're looking at the diffusion of the solvent instead of the solute. We're looking at the movement of water with solute dissolved in it. And that causes some wrinkles in ordinary diffusion as a result of looking at the solvent moving rather than the solute. First up, because we're looking at the solvent rather than solute, we're going to require a selectively permeable membrane. Remember that diffusion doesn't require a membrane, but osmosis does require a membrane. According to our rules of diffusion, we know that osmosis is water moving down its concentration gradient, so the big question here is how can you tell which side of a membrane has a higher concentration of water? In order to understand that, you have to remember what happens when a solute dissolves in water. If you look at the center here, we have a water molecule. It's a polar molecule, and that means that the oxygen has a partial negative charge, and the hydrogens have a partial positive charge. That's part of what makes it a great solvent. And so when you dissolve something like sodium chloride into water, that ionic bond is disrupted, and spheres of water molecules orient around each ion, such that with the positively charged sodium ion, the negative end, or the partially negative end of water, orients towards that big positive charge of the sodium ion. And you get these spheres of hydration. You have to picture this in 3 dimensions with water molecules forming a sphere that comes out of the screen and into the screen, so that all 3 sides surrounding that sodium ion. The same is true over here with the chloride ion that's negatively charged, except the water molecules are oriented so that the partially positive hydrogens are oriented towards that chloride ion. And because of these spheres of hydration, that keeps those 2 ions separated and keeps them from reforming their ionic bond together. The other thing that's true about this, though, and this is the important part, is that these water molecules that are attracted to these ions, these spheres of hydration around each ion, those water molecules are attracted to them in such a way that they can only go where those ions go. As you'll see in a minute here, that constrains where these water molecules are free to move. Here we have a similar model to what we saw in the diffusion video with some very important differences. We have a tank of water with solute dissolved on either side, and we have a selectively permeable membrane dividing the tank of water into 2 equal halves, so that there's an equal volume of water on both sides. That means there's an equal number of water molecules on both sides. But this selectively permeable membrane is very different than what you saw in the diffusion video. This one will allow water to cross, but not solute. Okay? That's the exact opposite of what we saw in the diffusion video. When you look at these solutes, you have to ask yourself, okay, if there's an equal number of water molecules on both sides, how is there a gradient down which water will move? To answer that, think back to those spheres of hydration around each of these solute particles. So remember that those water molecules around these solute particles can only go where the solute particles can go. And by definition, they're not allowed to cross the membrane. So those water molecules aren't allowed to cross the membrane. So on the right side, you have two solute particles with their spheres of hydration around each one. And the rest of the water molecules here they're free to move across the membrane. On this side, you have many more solute particles with many more spheres of hydration. So many more water molecules are constrained from crossing the membrane. Therefore, you have a higher concentration of free water molecules on the right side. And according to our rules for diffusion, that means water will move down its concentration gradient from right to left. That's the basic idea here, but there's another wrinkle because we're looking at the movement of the solvent rather than the solute. And that is this. When water crosses the membrane, that changes the volume on each side. So in our first example here, as water moves across from right to left, that increases the volume of water on the left side and decreases the volume of water on the right side. Ultimately, that means the left side becomes more dilute and the right side becomes more concentrated. Ultimately, what that means is that you can reach a concentration equilibrium sometimes where the concentration of free water molecules is equal on both sides, at which point there would be no more net movement of water. But as water moves across the membrane, it has to push water up against gravity and against air pressure, which we call hydrostatic pressure. It's pushing down on that column of water. The movement of water from right to left is osmotic pressure, pushing that column of water up. It's opposed by hydrostatic pressure, pushing down. So there's actually two different types of equilibrium that can be reached in a model or a situation like this. The first, if the initial concentration gradient is small, then you're likely to come to a concentration equilibrium. In other words, osmotic pressure will be sufficient to dilute the solute on the left to make it equal to the concentration on the right. If, however, the initial concentration gradient is very large, water will move over to the left, but it will stop moving when hydrostatic pressure equals osmotic pressure, regardless of the relative concentrations on each side. There's two ways you can come to equilibrium in a situation like this. To put this in more of a biological setting, think about organisms that have cell walls, like plant cells have cell walls and bacteria have cell walls. If you were to place a plant cell in pure water, okay, there's solute dissolved inside the cell, but no solute outside the cell, in the pure water around the cell. So osmosis is going to tend to move water into that cell from an area of high concentration of free water molecules in pure water. Clearly, the concentration of free water molecules is less inside the cell. So water is going to tend to move into that cell, but it's limited. Because of the rigid cell wall, at some point, osmosis won't be able to push water into that cell any further. And that's because of the hydrostatic pressure formed by the constraints of the rigid cell wall. If you think about it, the actual concentrations can never be equal because pure water, no solute. There will always be solute inside the cell regardless of how much water enters it. So they can never come to a concentration equilibrium. However, at some point, the rigid cell wall pushing back against osmosis will stop the influx of water. In the same way as the diffusion equilibriums, water doesn't stop moving across the membrane. It's just the net movement of water stops across the membrane. And that can either be because the concentrations have become equal or because osmotic pressure equals hydrostatic pressure. That's another big difference between simple diffusion of solutes and diffusion of solvents. Let's look at a more detailed example here. You know that molarity is the number of moles of a given substance in a liter. And a mole is a description of number of molecules or number of atoms. Osmolarity, on the other hand, is the number of moles of dissolved particles of any kind per liter. Let me show you what I mean. So if we take 1 mole of sodium chloride, okay, that means that we have Avogadro's number of sodium chloride molecules, that's 6.02 times 10 to the 23rd approximately, and we dissolve that in water, sodium chloride dissociates. So now we don't have just 1 mole, we have 1 mole of sodium ions, but we also have a mole of chloride ions. Similarly, when you put a mole of potassium chloride in water, you get a mole of potassium ions and a mole of chloride ions. So the molarity just talks to the number of moles of sodium chloride in that liter of water. Osmolarity looks at the total number of dissolved particles per liter. And so we refer to this as being 2 osmolar because you have a mole of sodium and a mole of chloride, or a mole of potassium and a mole of chloride. In this particular situation, you have equal number of total dissolved particles, and it doesn't matter that this side has potassium and this side has sodium. We don't care about the individual solute particles in osmosis because, again, we're looking at the movement of the solvent, not the solute. So it doesn't matter what those particles are exactly. It just depends on what the total concentration of particles is. And in this case, because the osmolarities aren't equal on both sides, this is already at equilibrium and there's no net movement of water across that membrane. Let's look at another situation that's, again, biologically relevant. Instead of using just ionic compounds like sodium chloride and potassium chloride, what happens when you dissolve a mole of something like glucose into it? So glucose is a covalent molecule, not an ionic molecule, but it still readily dissolves in water. Any polar molecule, whether it's covalent like glucose, many proteins, macromolecules are polar and dissolve readily in water. Just because it's not ionic doesn't mean it doesn't dissolve. What it does mean is that it doesn't dissociate, which means the sphere of hydration around this molecule is around the entire molecule instead of around individual ions. So when you put 1 mole of glucose into solution, it doesn't dissociate, so it remains 1 osmolar because you still only have 1 mole of dissolved particles. 1 mole of potassium chloride, though, when dissolved, does dissociate, so you have a mole of potassium and a mole of chloride, making it 2 osmolar. Now, let's talk about what we're going to do this week in lab. So, you're going to have 5 different solutions of known osmolarities. And what your job is, what you want to hypothesize about is, you're going to have 2 different potato types, sweet potatoes and regular potatoes, and you want to hypothesize about which potato type has the higher osmolarity. So to figure that out, we're going to have to determine the osmolarity of potatoes. So you're going to cut them into little potato discs. And then you're going to submerge them and incubate them in these 5 different solutions simultaneously. And the question is, how can you use osmosis to determine the osmolarity of the potato discs? And therefore test your hypothesis of which potato type has the higher osmolarity. Or maybe you'll hypothesize that they have the same osmolarity. I don't know. That's up to you. And just because I'm a nice guy, here's a little hint.